Okay, so one class of postsynaptic receptor is what we call ionotropic receptors. Now, in in any field, there are many different sort of terms that are used to refer often to the same kind of thing. So let me just describe what an ionotropic receptor does structurally and functionally at a synapse, at the postsynaptic membrane. And then I'll give you the other term that is actually often very, commonly used in laboratories and in research publications in addition to ionotropic. So first of all, here's our, presynaptic neuron. Here's our postsynaptic neuron, right? And we're going to see that, presynaptically, this neuron is going to release a neurotransmitter. Let's say it's the most common excitatory neurotransmitter in our central nervous system known as glutamate. And glutamate is going to traverse, be driven out of the vesicle presynaptically, right, upon fusion at that presynaptic membrane. Remember, that's exocytosis. The concentration gradient force will drive it out into the synaptic cleft, and then it's going to encounter receptors that are embedded in the postsynaptic cell. So this is the dendrite of the next neuron now that we're looking at here. And in that phospholipid bilayer, you've got these protein machines, these protein receptors, and they are, they go through the entire membrane, and before the binding of glutamate, we're going to see they are actually impermeable. There's no hole or gap or pore or channel to allow for any ions to pass through, right? But then glutamate binds, and then you see there's an opening of a channel, from one side of the membrane to the other. And for glutamate, this particular, ionotropic receptor will open a channel that will let ions that can depolarize the next neuron go through. And that includes, for example, sodium. So we're going to see there are specific kinds of glutamate ionotropic receptors that are called Ampa receptors and kainate receptors. Those are two different kinds of glutamate receptor. Ampa actually binds, is a chemical that binds specifically to the Ampa subtype of ionotropic glutamate receptor, while kainate is another chemical that can sort of chemically distinguish. It will only provoke a response in the kainate variety of postsynaptic glutamate receptor, right? And these two, the Ampa and kainate glutamate receptors, will actually open holes or channels that let sodium go through. And what I want you to think about is if you open holes to sodium in the postsynaptic cell, which is at the resting potential of minus 65, remember all of the sodium is outside extracellularly because the pump kicks it out. The membrane's not very permeable at the resting potential to sodium ions, right? So the electrical force and the concentration gradient force are going to drive sodium from the outside in, and that's going to do what to the polarization? Here, think about it for a second. It's going to depolarize. So you're bringing positive ions from the outside into the negative minus 65. So you're going to, basically depolarize towards the threshold minus 55, right, for, the initiation of an action potential, the opening of all those voltage-gated sodium and potassium channels. So glutamate acting at ionotropic receptors postsynaptically can rapidly depolarize, the next cell. And that's the advantage of these ionotropic receptors. They can rapidly change the charge difference across the membrane because they, the binding of the neurotransmitter to the receptor opens a channel or a hole. Glutamate has another very important postsynaptic receptor target that we'll talk about later in the class called the Nmda receptor. And Nmda is another chemical, that'll bind to glutamate receptors, but only to these special receptors that we call the Nmda receptors. And they have a different hole. When they open, in response to the binding of glutamate or to the binding of Nmda in the cleft here, they'll open a hole that will let calcium and sodium go through it. Calcium is pretty critical because remember, calcium, is a powerful metabolic actor inside the cell. It can initiate lots of intracellular changes, including, we will see later, changes in the expression of genes, like which particular receptors that will this neuron actually, build in the future. And we're going to see one of the ways that drugs can have a significant impact over the connectivity of neurons, the strength of synapses or the presence of synapses, et cetera, in your brain. So I want you to think about these ionotropic receptors, though. You can not only depolarize, right, by opening channels, by having the transmitter bind outside the cell and swing open a hole for sodium or for calcium, which will, are positive ions that will be drawn into the cell and reduce the polarization, reduce the charge difference, depolarize, and move that cell towards the threshold potential of minus 55, where you can generate, a full-on action potential or a postsynaptic, sort of excitatory postsynaptic potential, they call it. But you could also have, neurotransmitters that bind to ionotropic receptors that swing open channels that let potassium, pass through them. Well, potassium, remember, is in high concentration inside the cell at the resting potential because the pump has brought lots of it in. And remember, we said earlier that, potassium at, the resting potential, minus 65, if you open more holes to potassium, the concentration gradient force will win. It's stronger than the electrical force at that level of polarization at minus 65 at the resting potential. So the potassium, this positively charged ion, will leave the cell. And so you're moving more positive ions out. So you will be increasing the charge difference. You'll be making it more negative inside as you move positive potassium from the inside out. So you will be hyperpolarizing. And this is inhibitory. You're taking that cell away from the threshold potential of minus 55, which is a depolarized potential. So neurotransmitters that bind to ionotropic receptors that open potassium channels are going to lead to the exiting of potassium and increased in polarization, hyperpolarization, and inhibition of the cell. We're going to see there's other kinds of ionotropic receptors, right, that are going to actually open holes that will allow for the passage of chloride ions. And chloride ions are critical. Chloride, remember, comes in with sodium, so it's part of salt, and so it's in high concentration outside the cell, very high concentration relative to the inside in a neuron at rest. So if you open channels suddenly to chloride passage, well, that concentration gradient force at the resting potential, minus 65, will win again. And so the chloride ions, if you open holes in an ionotropic receptor, because the neurotransmitter bound out here, swung open the channel, and it lets chloride pass through, chloride's going to go from the outside into the cell, and that is going to increase the charge difference because you're bringing negative chloride ions from the outside, the extracellular space, into the intracellular space where it was at minus 65 relative to the outside. So you're increasing the negativity inside versus the outside. So you're going to go from minus 65, let's say, to minus 75, minus 80. At a certain point, it's going to equalize, right? Because the electrical force is now, equivalent to the concentration gradient force. But you now have a hyperpolarized, segment of membrane there. And that is inhibitory. It's going to make it less likely to fire an action potential. You're moving away from the depolarized threshold for the initiation of an action potential, the opening of those voltage-gated sodium channels. That's minus 55. And here you're going when you open chloride channels. And there's a very important inhibitory neurotransmitter we'll talk about called Gaba, which when it binds to its ionotropic Gaba receptor, it'll open a channel that lets chloride pass through, and chloride comes in, and it hyperpolarizes and makes the cell less likely to fire an action potential. It's inhibitory. It calms down neuronal activity. We're going to see lots of drugs act at GABA receptors, kind of like GABA, including alcohol, including benzodiazepines, including barbiturates, to basically hyperpolarize and calm down neuronal activity. Now, these ionotropic receptors have another name. They are also known as ligand, and I apologize for these terms, but ligand is a fancy word for chemical. So basically, these are ligand-gated ion channels, right? Ligand-gated channels. And that's because here you got, this is the membrane. Like here, I'm inside. My head's the nucleus. And this is the extracellular space. And here's GABA binding here, right? GABA is a chemical. It's a ligand. It binds to this ionotropic or ligand-gated channel. And when it binds here, it ungates the channel. It allows for chloride to enter and then hyperpolarize the cell.